Hello and welcome to this edition of Agricbiotech and Food Security, our weekly show where we delve into the intricacies of biotechnology as it concerns agriculture and food security. And today we shall be escalating conversation on what is known as gene editing. A gene is the basic physical and functional unit of heredity transferred from a parent to an offspring and determines characteristics of the offspring. So today we will direct our attention to gene editing, which is the ability to make specific changes in the DNA sequence of living organisms using enzymes that have been engineered to forge specific sequence. This is a crucial aspect of agribiotechnology that requires explanations to further deepen the understanding of the technology in its entirety. The topic will be interrogated from different perspectives. Our resource person today is the Director General of the National Biotechnology Development Agency, Professor Abdullahi Mustafa. He would provide us with vital information on gene editing of certain crops that have been approved in Nigeria and so many other crops across Africa that have been engineered to specific DNA sequence. The question to ask would be, how would gene editing process bring about improved crops and subsequent food security in Nigeria? It is indeed good to have you join me on this edition of the program. My name is Kirian Umayo. You're welcome. And today on the program, we have the Director General of the National Biotechnology Development Agency, NABDA, Professor Abdullahi Mustafa. Professor Mustafa is a professor of bioinorganic chemistry. So you're welcome to the program.
Thank you very much, Kieran.
All right, today we are dealing with gene editing to see how we can unlock new possibilities for crop improvement and food security. In the previous edition of this program, you made mention of improvement of crop variety and what have you, and that will as well ensure that we have a food security in the country. Now we are going to some specifics. For instance, what is gene editing?
Well, thank you very much. Genome editing, I don't want to go into details. I told you that, but I want to tell you how a common man will understand it, or will understand what a genome editing is all about. You know, the science of biotechnology, you can manipulate the gene randomly and then make a finding of what you actually want. For example, in the normal biotechnology practice, you have to transfer the gene into the crop of interest or substrate of interest, and then by transferring, you keep on transferring until you make sure, maybe in the fifth generation, you make sure that the gene is completely transferred into the substrate of interest, for example, a crop of interest. So that is when you are sure that it is being transferred. And then you keep on going from one point to another to ascertain that there is no toxicity, there is no mutation, there are a lot of things that you check in the normal biotechnology practice. However, going to genome editing, you don't have to go into all this practice of transferring of moving. What you need to do is to go and target the gene of interest. Get to a specific point where you know this particular portion in the gene is carrying a particular character that you want to enhance, you want to remove, you want to improve, or any other thing. So you go directly to specific area and you edit, you add, or you remove. That is exactly a specific science. Directly going there, it saves time, it is more specific, and you know the result that you are going to get because you know that this specific area is responsible for specific this. I'll give you an example of a layman. I mean, if you wanted to change a color of a particular, maybe let's say you have okay, previously there was white carrot, then it was changed to orange color. So you can change the orange color directly from that orange to purple to blue when you go.
The same orange.
Yes, the same carrot. You can go specifically and then change the color. You can do that to bring about a different color of that carrot. Instead of, remember, the white carrot existed thousands of years ago, and then gradually it changed to orange naturally, and that is biotechnology. However, you can still continue using conventional biotechnology to change the color, but this target specific, you can just go straight to the portion of the gene where it is responsible for color to alter it so that it can contain, it can come out with different color. So this is how it is. So if there is a certain disease in a crop, if the crop is being attacked by certain disease, you know that the attack will go straight to that particular portion and affect it. But what you need to do is to now alter that particular portion so that anything that is attacking, if it comes to that specific area, there is no portion, no level for it. And then it will go back and you have already solved the problem. So instead of wasting maybe 10, 15 years using conventional biotechnology, now you have a target specific biotechnology area, which is genome editing, so that you can do that editing and then you quickly get to result. So there are a lot of researches that are going on.
When you say gene, are you talking about the nucleus of the produce, or is it different from gene, nucleus and gene?
The gene is inside. It's the basic building block of life. Gene is the basic building block. So you find it in each and every.
Then what is
human.
the nucleus of, or do you also tamper with the nucleus of such crops?
Well, gene is in the nucleus. Just take it like that.
Okay.
You understand? Just take it like that for the conventional this
Okay.
thing. So it's inside the cell, but gene is responsible for each and every characteristic of anything, living organism, is contained in the gene. You understand? So if you're transferring the characteristic, it's going to be, the gene is what is transferred from one particular organism to another. That is what is transferred into. The character of this particular organism is going to be transferred. For example, I give you that explanation in the previous program that drought resistant or insect resistant or whatever is a particular gene that is being extracted. You understand? When you say Bt is Bacillus thuringiensis, you extract that particular gene and then you insert it into a crop. And when you insert it into a crop, that crop is going to be repelling the insect that will come.
Yes, because it's a combination now.
No, is that particular gene, the insect cannot, it doesn't, it's just repel the
Because
insect.
it's more fortified now.
No, it's not
Or
fortification. You see, for example, you see a food. The moment you see it, your conscience is going to tell you, this food I can eat. And when you see a poison, your conscience is going to tell you, this is a poison, I will die when I eat it. Exactly the same thing. So when you insert that gene, the insect will know that when it eats that thing, it is going to die. You understand?
Not eat it.
So it will not eat it.
So what people are really afraid of is it's safety, really. You know, some say it could be cancerous. Some say it could cause obesity. Some say many things attributing to GT products. But from all indications, from what we have had across the globe, Without BT, I'm not sure we're going to have food security in the country that is growing population-wise on daily basis, and there is no known birth control measures operating in this country and even elsewhere in Africa.
What I'm telling you here is that all these people that are speculating, they don't have proof. We are scientists. What we know is that if you have a proof, you bring your scientific finding, you publish it. People should see it and assess it, and making sure that, and to make sure that they can now replicate that experiment that you did to see whether they can get that exact result. If at all you cannot provide a proof scientifically, if at all you cannot bring physical evidence, if at all you cannot convince with a data something had happened somewhere, then all these are speculations, all these are fake news.
BT is where to go if you have to grow your agriculture, because population explosion is not impossible in a country where there's no form of birth control. What we have, some persons have a family of 24, just from one man, and that tells you that in the next, maybe by 2030, when we're looking forward to sustainable development goal attainment, it would have been up to 270 million people. And so on that note, we conclude this segment of the program, and I appreciate once more the Director General of the National Biotechnology Agency, Professor Abdullahi Mustafa, for giving us his time, of course, explaining from professional point of view on this topic. Thank you, sir, for coming. Thank you, sir. Also featuring on the program today is Professor Chidozie Egesi, Executive Director, National Roots Crops Research Institute, Umudike, Abia State. This is introducing a new TV series on agricultural biotechnology, one of the most complex and accurate scientific discoveries designed to provide sustainable food security for humanity. In the light of rapid population growth, Agricbiotech must be understood, accepted, and embraced to realize its full potentialities. Nigerian agriculture is vulnerable to climate change with associated occurrences, reducing agricultural production across the nation. But biotech offers new tools for increasing agricultural productivity and protection of food crops. And from farmers, testimonies abound on the benefits of genetically modified crops. In this TV series, we shall be dissecting issues surrounding Agricbiotech with experts on its understanding, acceptability, and application, week in, week out. Join me, Kirian Umaya, on our show, Agricbiotech and Food Security. Be my guest, Tuesdays, 7:30 p.m. on NT International. Brought to you by OFAB Nigeria, under the auspices of NAFDA.
Sir, can you just tell us what gene editing is all about? That's unlocking that aspect of technology for food productivity.
So gene editing is a new technology that is based on science, and is a matter of changing the DNA composition of any organism, including plants, animals, and even humans. So it is a technology that is used to do a targeted insertion of a gene of interest into an organism. In our own case, it's usually plants, and we have for cassava, for example, where we are doing gene editing at the high level. So it is targeting a portion of gene to either delete it or to add it. To delete it from the genome composition, and this is a lot of science, delete it from the genetic makeup of an organism. And so you either delete it and leave it deleted, or you delete it and insert a new one that you want to produce a new effect. So gene is a part of the body composition that determines how an organism behaves or functions. Let's keep it.
Okay, give us all we need to know about gene editing and the process of achieving it, plus its advantage over.
So It's a laboratory process where you start with the plant, you get what you call the explant of the plant. That is, go to the plant, the young portion of the plant. I'll use cassava as an example so that I can create an image. You go to the younger portion of the plant, you cut out the axis, the top. I wish I had things to demonstrate here for you at this time, but you can also go to our lab and we would showcase that practically. So you cut out that part, you put it in a medium in the lab, and you incubate it for a period of time. It grows in the tissue culture. We have a tissue culture lab. That's where we execute that. And you're able to use it to form what we call callus. That is a small mass. At that point, we can do the whole insertion of gene and our addition of gene, let's put it that way, where you can either insert, like I said before, or cut out. And after you do that, you now go through some processes in the lab that will lead to tests where you will show, is it where you demonstrate that it's showing that your results, that your effort was were successful, that you successfully edited the gene. So if you're able to do that, once you're able to show that, then you go to the field to demonstrate that. So we have an example for cassava bacterial blight. Cassava bacterial blight has no natural resistance. Almost all the varieties succumb to the disease when they meet the very virulent form in the field. So we had to find a way to engineer resistance into the plants with something we call sweet gene. And with this sweet gene, we'll be able to find where it binds to the DNA of the crop, and we're able to express resistance to disease. So we have a case of resistant cassava to bacterial blight because we edited it with gene, able to release them to Nigerians.
So what's the advantage of unedited version?
So unedited version, like I said earlier, does not have the resistance. In this case, in this cassava case, it doesn't have resistance to that disease. So whenever you plant your cassava in the field, especially where there are years that is very bad for cassava in terms of bacterial blight, you will see, you go to the field, during rainy season, the peak of rainy season, you will see all the leaves dropping like somebody poured hot boiling water on the leaves. These ones that are formed by gene editing show resistance, so they do not go through that disease. And that's just one example. There are other examples like drought tolerance, not in cassava, but in other crops like maize.
That has been Professor Egasi of the National Root Crops Research Institute, Umudike in Abia State. Next is Mrs. Ihoma, who will give us a demonstration of gene editing in a laboratory.
So what we do is, for example, this is a DNA molecule, and this DNA molecule contains the genetic materials that we want to use to achieve the genome editing. So the editing has already happened in this chemical that you see here. So the next line of action is to now put this chemical into your plant. For example, what we are working on is on cassava. We are trying to make cassava resistant to cassava bacterial blight disease using genome editing. So the target is to take this molecule and put inside this cassava. So now what we do is there is an intermediary step whereby we produce what we call calluses, and this callus you produce from here. From here you produce this. Now this makes it easy for you to, with this, you can now put, where's the bacteria? Okay, so you now, with this, you put it inside bacteria, then with this bacteria, you transfer to this, and from this, you recover this, and this goes to the field. So that's the practical aspect of genome editing.
This is introducing a new TV series on agricultural biotechnology, one of the most complex and accurate scientific discoveries designed to provide sustainable food security for humanity. In the light of rapid population growth, Agricbiotech must be understood accepted, and embraced to realize its full potentialities. Nigerian agriculture is vulnerable to climate change with associated occurrences, reducing agricultural production across the nation. But biotech offers new tools for increasing agricultural productivity and protection of food crops. And from farmers, testimonies abound on the benefits of genetically modified crops. In this TV series, we shall be dissecting issues surrounding Agricbiotech with experts on its understanding, acceptability, and application, week in, week out. Join me, Kieran Umaya, on our show, Agricbiotech and Food Security. Be my guest, Tuesdays, 7:30 p.m. on NT International. Brought to you by OFAB Nigeria under the auspices of NAFDA. We have been talking to a cross-section of Nigerians on their understanding of agri-biotechnology and its application, and whether or not the technology can assist Nigeria in food production.
Okay, I think with the increase in population and the scarcity of food, that would be the best route Nigeria should take, because biotechnology developed crops would produce more in lesser time, which is what would solve our hunger problem.
Okay. Nigerian government has declared a state of emergency on food and some policy markets. Are they the views that biotechnology's application is what we need?
The increase in population has led to eventual increase in hunger. Like, there are more people that need more food, meaning we need to find new ways to provide more food in less time.
What do you think are the advantage of biotechnology?
It's improved production.
Yes.
It can be used in prevention.
Yes.
And it can also be used in growth of things. So anything biotechnology is good when it applies to the content of the product. If it's in agriculture, like agricultural growth, you are applying biotechnology maybe in terms of chemical to apply it for the growth of crops to be multiplied. So I agree to that. The advantage is that it brings more food production and it promotes more agricultural products in Nigeria. Like, there are some crops that they grow in Nigeria that need biotechnology to grow.
In a world that has been crippled by a whole lot of things like Ukraine crisis, food security, or all of the COVID pandemic, I think we need something hasting in order for us to be able to generate so much food. So I think biotechnology is a very nice approach to that. I think we need something that can feed, like Nigeria now, there are over 200 million people. I think biotechnology is a very big concept that can help with that. So yeah, it goes. I think it's a very
some will argue that biotech is not good for our health. So what do you think about it?
A whole lot of things is not good for our health. Like, I think this is the 21st century and we are exposed to a whole lot of things, exposed to radiations.